All right, it is my great pleasure today to introduce Dr. Mark Geyer. He's a distinguished professor of psychiatry and neurosciences, emeritus, at the University of California, San Diego. He's been at the forefront of psychedelic research into psychedelic drugs for at least 50 years, and at least initially in pursuit of animal models of schizophrenia. Dr. Geyer, welcome.
Thank you.
So I think that you are in terms of the people I've interviewed for this series of Psychopharm Friday interviews, I think you're the record holder in terms of the number of papers you've had in the journal. I counted 69, incredibly nice. Your second paper was on the effects of LSD in rats in 1979 in the journal, for example. And I was wondering if you could just tell us more generally, what made you interested in psychedelic drugs as a pharmacologist and neuroscientist?
Well, psychedelic drugs made me interested in psychedelic drugs, if you will. And of course, I wasn't yet a neuroscientist. The term almost had been invented, neuroscience. There was a neuroscience research program. I think that was the first kind of formal use of the term neurosciences. But what prompted me to explore psychedelic drugs personally was a lecture given by Aldous Huxley at the University of Oregon when I was an undergraduate student. And it was the last year of his life, in fact. We didn't know that at the time, but I think he knew that he had cancer. And he came to the University of Oregon and he presented a very provocative and stimulating lecture that addressed a couple of interests of mine. One was then, the commonality of beliefs within the world's religions. And he wrote a wonderful book called The Perennial Philosophy, which very few people have the patience to muddle through. And a much more provocative work called The Doors of Perception. And it was toward the end of his life, and he was very much invested in psychedelic experiences. As you may know, he passed away later that year. April was when he gave this talk. He passed away in November, same day as John F. Kennedy was assassinated.
Wow.
And on his deathbed, he asked his wife, Laura, to give him an intramuscular injection of 250 micrograms of LSD. And he thought that was so nice. An hour or so later, he asked Laura to give him another injection of 250 micrograms of LSD. So these things, some of it was adolescent interest in mysticism and religious experience. I was a biology major with a philosophy minor, pre-med, and so I decided, well, I should try some of these experiences. And the only thing I could get was morning glory seeds, which tasted horrible, and you barely felt any psychedelic experience.
And they have LSA, which is like related to LSD, right? Basically, chemically.
Yeah, they have a lysergic acid. Albert Hofmann finally did identify that as a presumed active ingredient. Curiously, it's only some varieties of morning glories, and they're curiously named Heavenly Gates and Heavenly Blues and Pearly Gates. I don't know where the botanists come up with their names, but
I could guess. I actually.
At any rate, that and then psychedelics were still legal. LSD was legal. We could call it a rediscovery company and order whatever we wanted to, which several of us did. And I decided I wanted to study the brain because I was very impressed with the mental phenomena occasioned by the ingestion of even tiny amounts of these chemicals, which got me wondering about brain biochemistry and what errors of metabolism or catabolism might lead to in terms of psychiatric phenomena. And so I dropped out of my pre-med program and I was in the honors college there. You had to do a thesis. So I did my honors thesis on the behavior of rats in a maze after we were manipulating the hippocampus with bilateral cannula that I had to fashion myself. myself, actually. But so then I went on to um, get degrees in psychology, a master's at Iowa, which was a problematic time in the United States and in my life, 1967, 68, 69. That didn't go well. So I left academia and went to Berkeley and became a hippie, and decided, well, that life wasn't working out too well either, although I certainly met some interesting people. So I decided I'd go back to graduate school, and I applied to graduate school from a phone booth on University Avenue in Berkeley, and decided I'd come down to San Diego, where I still am, and work with the young 34-year-old chairman of a department of psychiatry that was less than a year old, because he had identified an enzyme in the mammalian brain that could convert serotonin into bufotenin, which is a relative of DMT, and which is one of the active ingredients in the ayahuasca sacrament. Um, I didn't know all that then, but I was interested in the hypothesis that errors of metabolism might lead to psychiatric disorders. It's one thing to take a drug and have these experiences, and the source of the experience. I took a drug, and that's why I really see these colors or feel these things, or but you didn't have the drug explanation, and you had those experiences that the world was there.
So did you get the impression then from those personal experiences that you were sort of, the drugs were making you enter a state that was in some way qualitatively similar to psychosis? Was that kind of like your impression? Because I guess, like, I'm asking because this, I think, is a motivation of a lot of your at least early research on this topic. Or did you feel like it was that was sort of one of the aspects of the drug experience, or maybe there was some overlapping experiences or something like that? Like, what was your take on that? What got you interested in the schizophrenia angle in particular?
Well, the identification of particular diseases didn't jump right out at me, but I think what prompted me to think about things psychiatric was this realization that if I had these experiences without an explanation, and they didn't go away when the drug wore off, in other words, that it would be very difficult to get along in society. I I enjoyed the psychedelic experience. So it wasn't that I, was terrorized by them, although some people do feel terror, especially if they don't know they've been given a drug. And many people do get rather anxious when one loses a sense of ego and self-identity. Not everybody tolerates that loss of control, loss of identity well, shall we say. It's now the domain on the questionnaires called AYA, anxious ego dissolution. When the ego dissolves, some people get a little nervous, more than a little.
Yeah.
So I mean, in a way, I didn't know anything about psychiatry. I didn't know about diagnoses particularly. The hypothesis that I read about early on was this notion that maybe serotonin, because serotonin and LSD and modern psychedelic use were born at about the same time in terms of their scientific life in the mid 60s or 50s, and so they were always hand in glove. And this hypothesis that the naturally occurring transmitter serotonin could be readily converted to a very potent compound, DMT, not DMT specifically because it's a 5-hydroxy or 5-methoxy version of DMT that would be in the brain if serotonin were the substrate. But I had the occasion to experience DMT. The serotonergic version of DMT wouldn't pass the blood-brain barrier, so you don't take that and get off unless you also have an MAO inhibitor. But DMT infused IV, injected IV, or intramuscularly, got to the brain rapidly enough that one has a 15-minute, 20-minute trip. It's very rapidly catabolized, so it's very short acting, but it's extremely profound. So, a couple of experiences with IV DMT when I was in that early experimental stage as an undergraduate got my attention. And put that together with what I came to understand as the biochemistry, the potential biochemistry, and the realization that without having an understanding that you're feeling these things, experiencing these things because of a drug, if this were the new me, made me very interested in this hypothesis that ought to be explored. Did not do that, but
So are you saying that at that time the hypothesis was, or a hypothesis was, that essentially the nature of schizophrenia was like DMT or something like that, that's like excessively high in the brain for unclear reasons and just uncontrollable, and that basically is psychosis?
yeah,
Oh
that
wow.
was a hypothesis in the early 70s.
Oh wow. Huh.
And there were very few studies done, of course. But Arnold Mendel, who was a young chair of psychiatry that was just starting out at a new university, a new medical school here at San Diego, later became a MacArthur genius. He was a very interesting, and still is a very interesting, provocative, sometimes infamous scientist. He had found N-methyltransferase, which is an enzyme that converts an indolamine into, it N-methylates an indolamine like serotonin or tryptamine, and thereby leading to the generation of a psychedelic compound, presumably in the brain. Now, the big obstacle to the hypothesis that one could explain a disorder, a lasting disorder such as schizophrenia, with a psychedelic endogenously produced, is the observation that there's very rapid tolerance to psychedelics, at least the serotonergic psychedelics. But there were studies and indications that maybe there's not tolerance to DMT in particular. And the jury's still a little bit out. I think it's probably, we know from very recent data that there is a very, very rapid short-term minor tolerance. Matthias Liechti and Basel have shown that there is even some tolerance within the time course of a continued infusion with DMT. But that's not enough, not like the tolerance that one observes and experiences with most serotonergic, all serotonergic hallucinogens or psychedelics. Mm-hmm. But there was one paper in 1976, I think, out of NIMH. Chris Gillen and Jed Wyatt did a study with IV DMT in healthy volunteers, given them every day, and every day they continued to rate it as the highest they'd ever been. Now that's probably a scaling floor effect because they used a pretty robust dose. So there could have been tolerance, they'd still be pretty high. But, so the jury was still out. Arnold produced a paper in 1975 in Pigeons that suggested, with mescaline, suggesting that some of the behavioral effects of mescaline did not show tolerance while others did. So in the 70s, this was still kind of a subject of dispute and debate. that, in my thinking and scientific career, I usually wasn't arguing that a disease like schizophrenia would be explained by that kind of endogenous metabolic error. But that my interest became understanding how these phenomena occur, even if they're not long lasting enough to explain a lifelong disorder.
So they may have overlapping features that could
Yeah.
explain aspects of schizophrenia, for example, or
Right.
psychosis or something.
So to make a living, you couldn't argue and make a living as a neuroscientist on that hypothesis largely because of the tolerance issue, and there were some other kind of red herring issues that I could more easily dismiss. But some of the phenomena were quite similar to some of the phenomena experienced and reported by patients in various psychiatric disorders.
Hmm. Interesting. So, like you're talking about a lot of this early work that you and others were doing in the 70s, which I think, at that time there was a considerable amount of research in this area, both with relevance to disease and I think just also on the drugs and on serotonin even in general, which as you mentioned is new. So, after that period, I think maybe late 70s and into the 80s and certainly in the 90s, the field kind of seemed to dry up from my perspective. Really vast majority of people, especially doing animal research, really just stopped. I'm curious if you could say something about that era from your perspective, like why that happened, and also then why that didn't happen with you, why you continued to be one of the few people that continued studying these things through that era during that, so to speak, dry spell.
Yeah, sure. As I mentioned, you couldn't make a living in the United States exploring psychedelics as a way to understand psychotic disorders or even psychotic phenomena. And that was because American psychiatry dismissed the endogenous psychotogen hypothesis, literally psychotogen meaning to generate psychosis. That's the original DMT bufotenin story. And so it was not exactly taboo to do such work, but it was certainly not, and it was a dismissed hypothesis in American psychiatry. So the only way we could get funding to do the research was to appeal to the drug abuse categorization of these compounds and get funding from the National Institute on Drug Abuse. So all of my interest was in understanding psychotic phenomena or abnormalities of consciousness and perception that may not necessarily be necessarily psychotic, but very interesting nevertheless. One could say psychedelic, which to me, those were not uninteresting phenomena, even if they didn't explain a disorder. But they explained something about the workings of the brain that was still, to me, quite interesting.
And was that the pitch that you sort of sent to NIDA, is that we're going to study these drugs, why are people abuse them, or what can they tell us about the brain, or...
Yeah. You had to couch it in terms of the dangers of these horrifically risky agents. And, society is suffering because they're out there and we need to understand them at a basic level in order to deal with it. How that was going to help us deal with it wasn't necessarily explained. So I had one grant R01 that lasted 33 years from NIDA on what was then called hallucinogens. We couldn't use the term psychedelics. And even though hallucinations are pretty rare,
right,
from the anyway, the legal definition was hallucinogens. And my last application for renewal of that grant at NIDA came back, and the major criticism, the major reason they did not want to fund this is, quote, what are we going to learn about addiction? even though nobody had really ever said they were addictive.
Except for the fact that they're Schedule 1, which is by definition um, addictive. But anyway.
No, addiction isn't part of the definition of Schedule 1.
Oh,
It's just
really? Okay.
abuse.
Abuse liability. Okay, I see.
Yeah. So it didn't require addiction, but this reviewer obviously thought that the purpose of NIDA was to study addiction, not to study the brain, not to study the phenomena associated with Schedule 1 or 2 drugs or drugs of abuse. I'm not the one who called them a drug of abuse. They did.
Yeah.
So it was their bailiwick. And, that argument had proven successful for a few decades, but finally, the acceptance of the non-addictive nature of them was our undoing, in a sense.
Wow.
So
Bizarre.
there are about 5 labs in the US that kept on through the dark decades. Dave Nichols, Richard Glennon, Barry Jacobs a little, George Agajanian, of course, and one of his protege, Mike Davis, and probably Jim Appel. So there were a handful of us, and almost all of the funding came from NIDA. Very little came from NIMH.
That's so interesting. I feel like maybe we're getting ahead of ourselves in the conversation here, but I feel like all of a sudden NIDA is again interested in this topic, but for the opposite reason, in that they're now maybe anti-addictive drugs.
Yes,
Very strange how the world works sometimes in terms of
it really is.
yeah. Wow.
And just to point out my reason to be explicit about in the US, because the other in Europe, psychiatry had not given up on the importance and potential of understanding the action of these drugs in terms of understanding psychiatric disorders or treating psychiatric disorders, especially Switzerland. Um, so in the 80s, well, no, it's very early 90s, I began working closely with researchers in Switzerland to do human work as well as animal, because I couldn't do human work in the US, nor was I trained to do human. Was not an MD, and nor was I medically trained.
Huh. Wow. So that's fascinating. I mean, thank goodness that somebody was holding the holding the torch throughout that period. As you alluded to, maybe starting in perhaps in part due to your work in the 90s, as you mentioned, and then certainly into the early 2000s, there's kind of been a renaissance, maybe you would call it, of this kind of research. Um, I'm curious if you could say a little bit more about kind of how that came about, like maybe some of the players involved or some of the key studies. I think some of them may have been in psychopharms. I'll mention that as well.
Uh, yeah, the turnaround I think was 1993. 1993, there was a little meeting in the south of Switzerland celebrating the 50th anniversary of Albert's understanding of the effects of this chemical that he had synthesized a couple of years earlier, the famous bicycle day experience.
LSD, Albert Hoffman, yeah.
Yep, Albert Hoffman. And so there was an invitation only meeting quietly sponsored by Sandoz, the company at which he had worked, to discuss and quote celebrate the Albert's discovery. And I was invited because I had done some published work with LSD, and so I was very delighted to be invited and go. And there was what kind of evolved during the course of the meeting was recognition that, well, boy, these Swiss and some German psychiatrists have been using LSD in therapy all these years, and we have no reason to believe it works. They've never done any real studies, and oh, my word, we better deal with that. I mean, we can't have that continue. So there was a challenge to what's called the Psycholytic Society in Switzerland, group of psychiatrists that used LSD to lyse the psyche and then rebuild it in a healthy manner. And of course, they're strong believers. They didn't really care didn't feel any need to do the studies, but the charge was put to them at that meeting kind of implicitly and but not somewhat explicitly that you better prove that this therapy works. And that was kind of in the preface to the book that came out from this meeting. For me, the meeting had a completely different effect because it gave me the opportunity to meet some people I ended up collaborating closely with. I met Albert and his wife Anita, and more importantly, I met Effie Gazoulis in Germany and Franz Vollenweider, Hermley, a number of others that with whom I developed collaborations, and so I began working with them, doing parallel animal and human research. And then the other thing that happened coincidentally in 1993 was a few of us in the US had been lamenting the fact that the only people doing psychedelic research were remnants of the hippie movement in the 60s. We were all getting old, we thought, and we knew when young people would approach us, say, how do you make a career out of studying these interesting compounds? You had to tell them, don't try. I mean, basically, there was not a career path that we could recommend. Even though we were on such a path, we knew how unstable it was. So you better have a day job. So notably, Dave Nichols was the leader of this conceptualization that we ought to try to do something to rehabilitate psychedelic science so that it might thrive when we retire. It might continue. Thrive was probably overly ambitious thought, but continue at least, because we were firm believers that these were interesting phenomena about understanding about their mechanism, and the phenomena could help us understand states of consciousness altered by whatever means, or just normal ecstatic states. So we started a virtual institute called the Hefter Research Institute, with the explicit goal to rehabilitate psychedelic science, get it back into the mainstream of neuroscience and pharmacology. And in order to do that, we had to have the highest possible academic standards. We had to publish in journals like Psychopharmacology to gain acceptance of this line of research. And we didn't have any endowment or money. We raised money from people who were, fellow travelers. Most of them had some experience. We had our first and very critically important first source of funding was one of the big leaders of Microsoft. Bob Wallace was about number 6 or 7 in Microsoft. And he knew chemistry as well, and he appreciated the science that we were exploring. So he was a really steadfast donor to us and to things like Dance Safe in San Francisco, the outfit that helps people identify what's in the drugs they've been given on the street. So Hefter raised money from philanthropists and accepted proposals from scientists around the world, or in some cases solicited proposals from scientists around the world, reviewed them rigorously using NIH-style reviews, external reviews, not just Hefter researchers.
Right.
But we worked much more closely than NIH reviewers with the scientists doing the work. And about 2 years ago, I think, the then president of the Hefter Research Institute calculated that I think it was 63% of the human papers published up to that point on psychedelics were not necessarily funded directly, but at least reviewed and funded in part by Hefter.
Well,
So Arthur Hefter was a classic German pharmacologist in the late 1800s. 1800s. He identified mescaline as the active ingredient in peyote cactus, and in the typical Germanic pharmacology way, the proof of the pudding is by tasting it. He had to try the extract that he produced from the mescaline that he produced from the cactus, and we got permission from his family to use his name. They were honored and pleased that he helped found a journal called Naunen Schmiederberg's Archives of Pharmacology, a very early German pharmacology journal. So it was a real joy for me to work with Franz Vollenweider in Zurich, and he's still a dear friend. I was at his house 6 or 8 weeks ago. We get together socially and as well as scientifically. In fact, we were the subject of a medical anthropology thesis on how one studies psychedelics in both humans and animals.
wow. Well, that's fantastic, because that's actually exactly where I would like to take the conversation next, is to ask you a little bit more about that sort of question, of how these drugs impact. Oh, here we go.
So this was his thesis.
Ah,
The revival of hallucinogen research since the decade of the brain.
there we go. Okay.
It was he visited our labs over a year, 2006, I think it was. So this was there was a revival, we thought, in 2006. He was an MD, a German MD, doing a habilitation thesis as a medical anthropology degree for his habilitation.
Wow. And 2006 is around, I think around the time of the first trials from Hopkins at in the US's, right? And
Yeah.
time, at least with psilocybin.
Right. Yeah.
Some of which were
pharmacology.
In psychopharmacology, exactly, by Roland Griffith and crew.
Roland, we brought Roland in after that we had worked with Roland in the generation of that, and a really interesting guy named Bob Jesse, who had encouraged Roland to do that work. And so Roland then became one of the directors of Hefter and worked with us throughout the next decade or more.
So then I guess my next set of questions is to do with, you being one of the most prominent people studying these drugs over the years in animal models. Obviously, there's some things that are very obviously appropriate to study in animal or even lower models like cell cultures and whatnot, met receptor signaling mechanisms and things along those lines, and this drug versus that drug and how it hits the receptors and things. But and this is something that I've been thinking a lot about as well recently, and as my own lab has started investigating these things in rats. There's some of the aspects of the experience that you mentioned got you interested in these drugs in the first place have to do with consciousness and things like hallucinations or at least changes in consciousness and perhaps sensory
perceptual distortions.
Perceptual distortion. Great, that's much better. Yeah. And so these are obviously things that are much harder to study in animals, and especially given that a lot of the studies that are out there now are looking at psychedelics as potential therapeutics, and there's this big controversy discussion about whether the sort of experiential effects of these drugs, like introspection and insights into past traumas and et cetera, things that one could imagine are fairly specifically human, or at least wouldn't be expected to occur in a rat that just gets injected with a drug and has no idea what's going on otherwise. I'm curious if you can tell me a little bit more about like what do you think about these animal studies? Are there things that are they're good for and things that they're simply just never going to be good for? Or are there ways that perhaps we could improve our models of even some of these more experiential kinds of things? I'm curious if you have thoughts on that topic.
Oh, all too many thoughts on that topic. But the dilemma is not that different from trying to study a psychiatric disorder.
Fair enough.
The idea of studying a schizophrenic rat, not quite sure we're ready to do that.
Yeah,
People
I don't
try
know what
to
that is exactly.
study depressed rats, thinking that they've done something to them to make them depressed. But so we're not in a unique situation with psychedelics to try to do cross species work. The thing in psychiatry in general, is to do cross species work, you've got to have some common terminology. And unfortunately, most of the definitions of psychedelic experiences or psychiatric symptoms are reported and assessed and studied using language. And, Trevor Robinson and I have had a lot of thought and work about trying to overcome this barrier of, all the stuff we're trying to study by studying the neuroscience of phenomena related to things psychiatric phenomena are the impediment. A fundamental impediment is all the human phenomena are expressed in language. All the
How do you know
clinical
if
trials use language.
how do you know if a rat is experiencing oceanic boundlessness, for example?
Yeah. And how do you know the human is?
Yeah, good question.
And in psychedelic work, you know what the most commonly used adjective to describe psychedelics is?
No.
Ineffable.
Ineffable. Okay.
So, it's like, well, you know it when you see it, but I can't tell you what it feels like. I mean, it really is hard to explain to somebody who's not been there what the experience is.
That's so fascinating. So would you say, I mean, maybe I'm just asking you for your opinion here, because obviously this is maybe not something that's empirically answerable, but would you say that the quality, like the experience of a psychedelic drug, if given to a rat or a human under similar circumstances, would be qualitatively similar? Or do you think there's something about the brains, the huge amount more cortex we have, or maybe the different organization that's curving over evolutionary time or something like that, that makes the experience just simply different? Or do you think they are fundamentally the same? It's just a matter of asking rats about it, which we can't really do. You see what I mean?
I think they're neurobiologically the same. I don't know if that's fundamental or not, but
Meaning they hit the same receptors kind of
yeah,
thing. But then what if, like, for
that's
example,
an article of faith. I can't prove that.
yeah,
But
sure, sure. And there's also like, even if they do literally mechanistically work identically, maybe the receptors are on different cells, or maybe they're in a layer of cortex that rats don't have, or whatever. There's that kind of thing. But then there's also the sort of next level kind of thing of like, it's affecting their consciousness or whatever, which, we don't quite know that they even can't even prove they have it, let alone measure it. So I guess just like so your sense is that probably they fundamentally are basically the same. Is that fair to say, or?
yeah, I believe they're the same mechanism. Serotonin is the earliest GPCR chemical known in biology.
Mhm.
It has been used by animals in a variety of ways as a chemical communicator between cells and among cells. I think so. I mean, I think the experiences have to be species specific. So the consequences of a change in dopamine or serotonin or acetylcholine are going to be elaborated differently in a species specific manner. A lizard might be affected by a serotonin agonist, but in a human is psychedelic, I'm not going to say you have a psychedelic experience in a lizard, but you have whatever the lizard system uses that communication signal to do, it'll do.
I see. So,
And
was it
as you
fair to
get
say-
closer, you up them, get into mammals anyway.
yeah,
You're going to be a little more close in terms of the experiential aspects of the species being similar, more similar to primates, and you know,
yeah,
so I don't think we want to or need to try to equate the phenomenology from a rodent to a dog to a infrahuman primate to us, the talking primate, you know, or I don't think anybody. John Lilly would love to have tried study LSD. He preferred ketamine to his dolphins when he was trying to learn to communicate with the dolphins. But somebody did kill an elephant with LSD, but I don't think anybody's looked at that other mammal that is at least arguably as sentient as we are, and is
exactly.
complex, an organism with communications that we don't understand, but whales and dolphins, one could argue, might well have a very elaborate life experience, communication, social network. It's supposed to be a phonetic language that they're now talking about for whales.
Mhm.
Others have speculated on holographic images projected for that huge sinus mechanism that they have that they could project. They can certainly, there's reason to think they can image each other. So, I mean, I don't know that anybody's tried a psychedelic, or I don't know much about the serotonin system in a dolphin, but that doesn't. So at a fundamental level, I think there are going to be some similarities, but they're going to be expressed in
Sure.
species specific ways.
And it's actually kind of like almost opens up a so when you're talking about rats and mice, obviously their sensory world, their umwelt or whatever you want to call it, is more dominated by things like smell and touch than humans, which is much more visual based. And obviously some of the sensory disturbances and things you meant that occur in humans are visual based, and one might have easily predict then that the same thing happens, but in the domain of olfaction, or in the case of dolphins, which, as I understand, don't really have olfactory bulbs, for example, or don't have like a developed sense of smell. Maybe it's in some
Right?
other
They have
domain.
chemical senses, probably.
Yeah, so maybe that's in a sense they do have smell in that way, or at least something
Yeah.
analogous to it. But yeah, I totally understand what you're saying. Like maybe it's this sort of same fundamental change in processing or something that occurs in all these brains, and it just kind of filters through the way those particular brains work in their own evolved environments. That
Right.
makes a lot of sense to me. And so one of the things that I struggle with when it comes to these studies that we're trying to do now in my lab, where we're looking maybe at potentially therapeutic effects, quote unquote, of these drugs is, we're trying to capture some of the aspects of the clinical trials that are occurring now in which people are maybe, let's say, depressed. They've tried a bunch of other things and nothing seems to work. And now they sign up for this trial in which they understand that whatever they're going to give them, probably they even tell them it's a psychedelic, it might make them better. And so they're going in with an expectation of therapy, basically, which is just kind of, to me, qualitatively different than what our rats have. Even if they are depressed, let's imagine we somehow are depressed in our models. They're not going to get the drug and know that this is supposed to make them better or something, and that might well be a factor that is important for the therapeutic use. So what do you think the prospects are of these kinds of studies in animals? Are there ways we can kind of get around this, or maybe it just doesn't matter? Maybe it's just simply hit the receptor in the right way and the good therapy, plasticity, whatever happens, and they're better? Or is that just a kind of barking up the wrong tree?
Well, I'm glad you mentioned that, because when you first asked the question, I thought, oh well, one of the big mysteries for me is, and sources of skepticism in a way for me is, how are we going to understand the mechanism, neurobiological changes that are occasioned by these drugs that are responsible for the therapeutic effects? There are going to be a lot of changes associated with the experience. that's subsequent to the experience. You lose a night's sleep. There are a lot of plastic changes. The brain changes.
Exactly. Yeah.
You remember what you had for breakfast? That's because there was some neuroplasticity, and it's expressed because you remember what you had
Probably
for
some
breakfast.
prefrontal cortex spines or something grew about that, you know.
Yeah. So that there are, changes in spines or what, connectivity. That doesn't mean they're the right ones. I mean, so I struggle with how you do conceptualize an experiment to pin down, even in a human, if you could sacrifice them. I mean, even if you could do real neurobiology in a human, or certainly with the neurobiological tools that we have now, which are remarkable, I must say, how do you conceptualize an experiment to capture the the subset of the many sequelae that are germane to the therapeutic change in the organism, the antidepressant change for depression? We've known for a long time and been quite comfortable with a single profound negative phenomenon called trauma causing a lifelong change called PTSD.
Yeah.
And yet everybody's just amazed that a profound afternoon on the psilocybin can have a long lasting change. What's the big deal? I mean,
Well, it's striking and compared to a lot of other drugs, for example, which I think in concept you're exactly right. I love that analogy that of course a trauma can cause a lot of legacy lasting changes due to plasticity. Why couldn't a drug do that? It's just that most drugs don't do that. Most drugs have an effect and then it goes away, and most of the time that's basically all there is to it. Whereas these drugs seem to, I mean, my personal hunch is that they're tapping into something to do with significance of an experience, and just making
really,
it seem like whatever it is that's going on in your head, maybe it's a therapeutic thing, maybe it's a bad thing, it could cause long-lasting trauma, even one might imagine, but whatever it is, it just sort of like tags that as a very important thing, and that's the kind of thing that trauma does as well, due to obviously very different mechanisms. But to me that's kind of one way to conceptualize this potentially.
absolutely. I mean, the first answer to that is, you say most drugs don't do that. Well, but these aren't most drugs.
Yeah.
I mean, the phenomena can be quite profound. The experience can be quite profound. Um, Opium, for instance, even after a single or two or three administrations, will lead to, is sufficiently impactful to shape behavior that then continues. But that's a different situation because the behavior that it's leading to is repetition of the same
Hmm.
experience. And there's a little of that with psychedelics. Many of us enjoy it enough to go back and do it again. Not in a pattern that could be called addiction. Very few people want to go back and do it tomorrow. Um, and if you do it every day, it stops working. So I mean, it's kind of
Self-regulating in some ways, eh?
self-regulating. Yeah,
Yeah.
but the argument that while other drugs don't do it doesn't carry too much weight for me because the other drugs don't have the profound, don't generate the profound experience that psychedelics can.
Huh.
Now, many people are arguing that we and many companies are banking a lot of money on the idea that maybe all you need is serotonin 2A agonism and you don't need the trip.
Right.
Wouldn't it be convenient? Because then we wouldn't have to monitor them, have clinicians and all this messy stuff,
Magic
costly stuff,
magic bullet is what we're looking for.
expensive stuff. We could just give them a non-psychedelic serotonin 2A agonist, work through some different transduction system or something, and sell a lot of drugs as therapies. My belief is that's not going to happen.
Hmm.
You know, a person that drives down the street and doesn't get into the accident doesn't have PTSD. Not even 1% of them that drive down the street and don't get in the accident.
Hmm.
You don't. I just don't think you're going to get there without the profound experience. It's not most drugs. They're a particularly impactful class of drugs by virtue of the nature of their experience.
Interesting. So then, given that we can't ask animals about the nature of their experience, and so it's really kind of inaccessible in most, probably for us to study that in animals, would you say that there's sort of some kinds of studies that we should be doing in terms of psychedelic research in animal models, and other types of studies that we just simply shouldn't bother because we're just never going to be able to do that?
Yeah, sure. I think right now to understand the which of the many sequelae that are probably going on are responsible for a therapeutic outcome, a change in some rating scale, or the whatever change occasions a person's ability to stop smoking. You know, there's a definable endpoint that nobody argues about, and yet the efficacy is amazing. In fact, Matt Johnson's work is very impressive.
Yeah.
Um, in the meantime, and I think the same thing is true for psychotherapeutics for other psychiatric disorders, for psychiatric disorders, like antipsychotics or drugs that might treat bipolar disorder. Um, I think we can understand and them neurobiologically by working in animals, but in order to assess the clinical outcome, you're going to have to do clinical studies. Now, what Trevor and I have both argued is that we would really benefit by having an improvement in human experimental medicine paradigm.
Hmm.
What he's done with Cantab, for instance, that's an explicit, planned set of tools to assess function, mostly at a behavioral level, in a nonverbal way, in enabling a more close relationship between what we study in animals and what we study in humans. And if everybody's blaming the preclinical animal models for the failure to predict the efficacy of psychiatric drugs in phase 3. But you'll notice we can't predict the efficacy of psychiatric drugs from phase 2.
Yeah. It probably is relevant that, what we're calling depression is sort of basically defined on something that's based on insurance companies and things, as opposed to
Right.
like the brain. You know what I mean? This is, what your endpoint is it effective or not, is like, does it affect one of 10 things that define depression in the DSM?
Right.
Maybe that's a problem. So I would agree with you that there's problems on both ends of the translational pipeline here.
And the same is for the group of schizophrenias. It's not a single disease. There's not a single etiology. There shouldn't be a single treatment other than at some common final pathway, which is whatever bothers society.
Interesting. Wow. And then I have just so many other questions I wish I'd ask you, but in the interest of time, there's one thing I really want to get your take on. So, as we've talked about, there was this initial burst of scientific interest in these drugs, I think starting in, with Albert Hoffman in the 40s and then up through the 50s and 60s as they kind of hit the mainstream and became scientifically studied a fair amount. And then there was this lull for a long time, and now it's come back. And so I'm curious if you could say anything about, in the context of history, the era that we're now in, on the kind of upswing of the potential exciting benefits of these drugs. A lot of people are all in on this. A lot of people trying to make a bunch of money on this and make people better with these kinds of treatments. Is there anything we can learn from the initial rise and fall that might be useful in terms of how the field progresses from here? Are there big like warning signs that you see? Is there anything else that we really need to, as a field, pay close attention to in order to mitigate some of the downsides or whatever it is that caused these the sort of downswing of research on this in the first place? What can we learn?
Well, one answer to part of that is kind of the same thing we undertook with the Heffter Research Institute, which is do better science. So a lot of the problem of the work and the enthusiasm of the 60s, where these drugs were perceived by some as panacea, we're seeing again now. And it behooves us to just do the most rigorous, skeptical science that we can do. So, I mean, that's, I think, one way to minimize the probability, the likelihood that we're going to have a repeat of what we saw in the end of the 60s when it was all shut down because it scared society, scared Dick Nixon. And so the warning signs that I'm worried about and perceive, it comes about in part by virtue of this enthusiasm, the hype. Michael Pollan's book was an important piece of work and had its value, but it also made expectancies and placebo effects all that much, and placebo disappointment effects. Not just the positive expectations in the drug group, but the disappointment in the placebo group because of the expectations. The enthusiasm that has led to so much investment concerns me because a lot of it is not being done by people who know what they're doing.
Yeah.
Not being done by people who understand what they're working. On some occasions, they have a profit motive.
Sure.
And they don't. I mean, many of these people have never had this experience. They don't know what they're suggesting their patients are going to be experiencing. And then the other consequence that we're seeing, which I have to give mixed reviews to, is decriminalization. I've never been much of a believer in prohibition as a way to solve the ills of society. So I don't know what I would, if I had control, and I don't, I don't know what I'd recommend that we do, because I do feel that these experiences, these drugs, are not good for everybody.
Yeah. And if you just buy them in the regular store or whatever, like I was at Society for Neuroscience meeting, I was in Washington, D.C., and I was walking from my Airbnb to the conference center, and there's like 5, 6 places that were just like, regular little like convenience stores or whatever. They were just selling mushrooms because it's decriminalized. It's kind of a legal gray area. And to me, that was, again, I'm not a fan of prohibition either as a way to solve drug addiction problems, but just handing these very powerful drugs to absolutely anybody, especially with the sort of implicit promise of like they'll make you better,
Yeah.
is to me not necessarily the best path either.
Because my concern for the field is that the untoward, unwanted, and the dangers, the outcome of administration consumption by people who shouldn't have it will be very unfortunate and lead to they're going to throw the baby out with proverbial bathwater.
That's what I'm worried about too. And I really feel like that partly happened in the past as well. I mean, Tim Leary was a scientist at Harvard and
Absolutely
doing legitimate science, and then he kind of like got high in his own supply, like literally and figuratively, and basically tune didn't turn on and drop out, so to speak,
Right.
which I don't think helped the field, although it, scientifically, let's say, at least.
His colleague Richard Albert, went to India. He came back as Ramdas, and he helped Hefter.
Hmm.
He was part of our support system for Hefter. I mean, two professors went different ways and had different effects on the world.
Interesting. So then, one other thing I've been thinking a lot about is, as a person who's like, studied addiction for a long time, has not been especially interested in psychedelics for their own sake. I don't have the same sort of personal history as you do. But now we're getting into it as a research topic. I'm sort of, when talking to people in the field, almost everybody I feel like has had personal experiences recently, are a little bit like on board from a personal perspective, and I'm trying to think like, would that be good or bad for me to use these drugs myself to try to understand them better when I'm in my studies in the brain? And I'm mixed. I don't know quite what to do, because I feel like there's, as you said, there's these drugs have really profound effects on how you think about things, and why not? The scientific topic is going to be one of those things if I personally use them. And I wonder if there's maybe a niche for the field to have somebody who is literally not invested, does not know about that stuff, and will come in with that maybe more skeptical view and so on. Or else maybe I'm just totally misguided because I don't have those. I don't know what to look for, or something like that. I could see arguments on both sides. And I'm not asking you for advice about what I should do personally, of course, but I'm curious if you have thoughts on, I mean, especially given your own trajectory and such. What do you think the benefits and costs are? If maybe there are costs, I guess one would say, of using the drugs yourself if you're trying to understand them scientifically.
Well, not quite directly answer your question, but the one domain where I think it's germane is in the training for monitors or
Hmm.
guides. We don't call them guides; we call them sitters or monitors, but they could be guides. I mean, maybe we're going to evolve into some psychotherapeutic intervention enhanced by psychedelics. Right now, that's a complex story, but I do think it's helpful for the sitters or the monitors to know what the subject is experiencing. And in fact, I think it's helpful for the subject to know that
Hmm.
the person who's advising them, if they're having troubles. So, that it's not a required part of the training for therapists, but I think it will help make therapists, even if they're not really doing formal therapy, know what the subject is experiencing,
Hmm.
have some appreciation.
Yeah, that makes a lot of sense.
I'm thinking this, depending on the nature of the questions you're asking, it would help the scientist design their experiments better. But if one wants to do cross species work, some of the work could be done very effectively without knowing what the psychedelic experience is. I don't think we have to be a schizophrenic to study schizophrenia.
Sure.
What we've done is to use nonverbal tests like psychophysiology, startle, simple information processing, or perceptual aberration processing, speed of processing kinds of measures that we can do in humans or in animals. We have mice are amazingly adept at some of the cognitive tasks that we use in humans. So, and humans can even do some of the cognitive tasks that we do and use in animals. We set up a human behavioral pattern monitor after studying exploratory behavior and locomotor patterns in animals. And we just decided to do the same thing. And sure enough, bipolar patients, manic patients look a lot like dopamine transporter knockout mice because they just explore the hell out of the world and they're disinhibited and make risky choices on Iowa gambling tasks. And so we can do non-verbally based human experimental studies, whether by drug or by disease, and makes some cross-species characterizations that I think have a measure of validity. I think if we were to put more of those kind of human tasks into phase 2 studies, we'd have better luck at identifying which kind of patient with what kind of symptoms needs what kind of drug.
Yeah, I think it's a great example of, many times us animal researchers kind of try to look at the human condition and design our things to be as close as possible to that, to try to study these disorders, et cetera. But I think there can be value in going the other direction too, as you just pointed out. Many cases of, we have animal models, that's what we do. It's what we know a lot about the neurobiology, and if we can put humans in a similar situation, maybe we can make some new insights as well. So, this reverse translation stuff, I think, can be really important as well. And I applaud your efforts, I think with Jared Young in particular, over the last several years to do that kind of stuff. And really, really fascinating stuff. And I guess I'm going to have to stop it here. We're already way over at the time that you're generously have given me an extra 15 minutes that you promised. So, we may have to do another sort of part 2 of this at some point. I have so many more questions about the animal models and things that I'd love to go into, but let's draw to a close now. And I just want to thank you again for your time, Dr. Geyer. This was really a fascinating conversation and really an honor to get to pick your brain about this important and timely topic.
Okay, well, thanks for thinking of me, and been fun to have the discussion.
My pleasure entirely. Thank you again.
Okay, bye-bye.